Thank you, Catherine. My name is Andrew Hipp, and I am the current president of ASPT, American Society of Plant Taxonomists, and now feeling a little flustered. On behalf of co-organizers Jennifer Mandel and Catherine Picard, as well as the entire ASPT membership, I'm delighted to welcome you to our second webinar of 2026. If you're not a member of ASPT already, we're particularly happy to have you and encourage you to consider joining the society. There should be a link to the ASPT website in the chat to learn more and become a member. Our webinar series this spring explores the question, what good is taxonomy? Each of the 5 talks highlights places where taxonomic work affects people well beyond the boundaries of research. ASPT believes that fostering a strong natural history community is more important than ever, and we hope that this webinar series helps to strengthen that community. Each webinar, including this one, will be recorded and posted on our YouTube channel. Catherine is putting a link to that channel in the chat as well. Please make a note of our next talk in the series, which is by Dr. Wes Knapp, Chief Executive Officer of the Center for Plant Conservation. Dr. Knapp will be speaking on the intersection of taxonomy and conservation. His webinar will be Friday, April 10th at this same time. And you can register at for that in the link that's in the chat as well. And I encourage you to do so right away so you don't miss that exciting talk as well. And now to the talk we've all been waiting for. We are honored to have Dr. Susan Pell as our speaker today. Dr. Pell is the Executive Director of the United States Botanic Garden, where she has served continuously since 2014. In her leadership role, Susan is dedicated to helping the garden achieve its mission of inspiring people to appreciate, study, and conserve plants to enrich society locally and globally, and to diversifying the collections, offerings, and visitorship of the garden. This is a vision she's honed individually over a career as a botanist, educator, and leader in botanic gardens. In addition to her leadership roles, Susan maintains an active research program. She has traveled the world to collect plants for scientific study. In addition to numerous peer-reviewed publications, Susan is the author of an illustrated dictionary of plant morphology, a botanist's vocabulary, which is aimed at naturalists and gardeners. She is also notably the 2020 recipient of the Peter Raven Award from our own society, the American Society of Plant Taxonomists. We are excited for Susan's talk today on the role of taxonomy in botanical garden education, outreach, curation, and conservation. Please join me in welcoming Susan Pell.
Thank you so much, Andrew. I'm really happy to be here with you all today. I'm excited to talk to you about the wonderful science that happens at public gardens. Right, here we go. So as Andrew said, I work at the United States Botanic Garden. And I think when people think of public gardens, a lot of people think of, just the beautiful plants that we grow. We're coming into the spring season here, and we're going to have our highest visitorship of the year coming to just see our wonderful spring blooms. People might think of rose gardens and, a stroll through a restorative space. They might think of the diversity of a particular type of collection, such as aquatic plants, and the ability that gardens have to display these in mass. A lot of people will think of gardens as places to come to enjoy an exhibit and enjoy perhaps a holiday show, such as the one here at the US Botanic Garden. They may think of the people who work at the garden, of the gardeners, and the immense skill that it takes to maintain living collections, and the use maybe of those collections for other purposes. People enjoy coming to gardens, and we sort of take advantage of the opportunity of having a very large visitorship here at the US Botanic Garden, over 1 million people a year, and inspiring those people to really care about plants. We do this through displays of beautiful plants, as you've seen, but also through engaging opportunities to teach people about the plants that are important to us in our daily lives and to the environment. There also is a lot of science that happens at public gardens, and I think many people that visit gardens aren't necessarily aware of that. I think this audience is probably very keenly aware of the science that happens at public gardens. Certainly the taxonomic work, a lot of field work comes out of public gardens. And a lot of scientific research goes into the maintenance of our collections and the use of our collections to gain more information about plants. So today I'm going to talk to you about the power of public gardens and using our sort of platform, both online and in person, to really engage people in science and engage people in learning more about the plants in our collections and the plants throughout the world. So I'll talk a little bit about the science that happens at public gardens and how we engage people through displays, through education, and through exhibits. So if you've been to a public garden in your area, you've probably been on a high visitation day. Maybe you've been on a low visitation day too. But public gardens receive over 132 million annual visitors. This is more annual visitors than go to NFL, NBA, and Major League Baseball games combined each year in the United States and Canada. Over 3 million school children participate in educational programs at botanic gardens. These are going to be almost entirely all about plants. We also transfer or donate over 600,000 pounds of produce from public gardens each year. And there are 128,000 acres that are under the care of public gardens in the US and Canada. And finally, I want to talk a little bit and the sort of power of gardens about the conservation impacts of public gardens. So globally, 30% of all living plant species are in living collections at botanic gardens. And this includes 41% of all threatened plant species within these collections. This is globally now. And most of these collections are ex situ, so they're under the care cultivation of experts at public gardens. But many public gardens also care for natural lands as well. So some of these collections are in situ. All right, so how do we utilize these to really advance scientific knowledge? There's a lot of different types of science that happen in public gardens. I'm sure that, as I said, you all are familiar with many of them. There's quite a few herbaria at gardens, many training programs from kindergarten all the way through graduate degrees and beyond at public gardens. So we're going to go through some of that. How do we facilitate this research, though? How do we facilitate science at gardens? Literally with our facilities. So public gardens have conservatories, very large greenhouses that are really designed for public display of plants. And of course, outdoor gardens as well. We also have, in many cases, support greenhouses. In most cases, we have support greenhouses. Some of those support greenhouses are dedicated to growing up display plants, and others may be dedicated to scientific research. This is our production facility, which is located just sort of south of our campus, and this is an 85,000 square foot greenhouse range here at US Botanic Garden. But public gardens also have scientific facilities like academic institutions do, where a diversity of types of science are conducted, including systematics and taxonomy, genomics, anatomical studies, and plant propagation, including micropropagation as well. And many of the world's largest herbaria are at public gardens. Each of these gardens listed here, World Botanic Gardens Kew, the New York Botanical Garden, and the Missouri Botanical Garden, have collections in their herbaria of about 8 million specimens. And I'll just note, too, that the largest herbarium in the world, which I'm sure you all are familiar with, is in Paris, and that's at a natural history museum. And I would sort of argue that public gardens and museums are really kind of in the same collection or category of cultural institutions. We have ordered collections that are accessioned. So a museum collection, like an herbarium, is very similar to a living collection, which is also accessioned in a database, et cetera. I'm going to go through a few example gardens. Some of them you're going to be very familiar with. The New York Botanical Garden. This is, it's one of the largest herbaria here in the United States, probably the largest herbarium in the United States. And they also have molecular labs. For a long time, they've been doing molecular research there in the Coleman Lab, which is focused on systematics research. They also publish directly several different peer-reviewed journals, or Britonia, and then they support the publication of other journals. They also offer graduate education programs through partnerships with universities in the New York area. And they host professional organizations that are related to taxonomy. So the American Society of Botanical Artists and the Torrey Botanical Society are both based at the New York Botanical Garden. Missouri Botanical Garden, also probably familiar to many of you. I'm sure that many of you have visited their herbarium, as have I. They have molecular lab as well. And they also publish many different scientific publications, and I'll show you some examples of those in a minute. And of course, for many, many years have sponsored and housed graduate students, and also have adult education programs in botany as well. And they host a number of international flora projects and have historically as well. Denver Botanic Gardens also may be familiar to you all, Desert Botanical Garden as well. Both of these have an herbarium, they have molecular labs, they do a lot of direct conservation work, ex situ and in situ, and they offer scientific education programs, mostly in plant science. And then there's a lot of other gardens around the country that maybe you aren't as familiar with. So Louisiana State University, where I got my PhD, has an arboretum. It's called Hilltop Arboretum. And they do a lot of science out there as well at a different scale. So they have done a plant inventory. They offer a lot of educational programs, mostly to elementary school students. They've done invasive species eradication efforts as well, in partnership with some other organizations in Baton Rouge. And they host a native plant sale every year where they get to educate people about plants native to Louisiana and encourage them to grow them in their gardens. There are over 600 public gardens in the United States, so our power is also in our numbers. There's just so many of us, and we all have different missions, but we're all growing plants, displaying them to the public, and teaching people about plants. Globally, there are over 3,000 public gardens, and about 1,800 of those gardens engage in science. So what kind of science are we doing at gardens? Well, we're doing taxonomy for sure. Taxonomy is something that people here at U.S. Botanic Garden and gardens throughout the world are doing pretty much every day. We're identifying unknown collections. We're often naming new species. We're contributing to floras and other such contributions. Just a few examples of some of the floras that have been generated at public gardens around the world. Most of the major public gardens have people or have press offices that contribute to floristic studies. Many gardens, as I mentioned, directly publish scientific journals. So Annals of the Missouri Botanical Garden and Novon are from Mobot, Kew Bulletins from the Royal Botanic Gardens Kew, Bretonius is from the New York Botanical Garden, and of course Edinburgh Journal of Botany is from the Royal Botanic Gardens, Edinburgh in Scotland. But these are just a few examples. There are many more examples of scientific journals, peer-reviewed scientific journals that are published out of public gardens. And then there are all of these other publications that come out of public gardens that lean heavily on taxonomy or help people understand more about the classification and naming of plants. And these include publications in horticultural science and natural history, and many reference books as well, such as plant glossaries and dictionaries. Just give you an example of sort of how we use taxonomy here at the U.S. Botanic Garden. So just one example of a single collection that really relies heavily on the taxonomic expertise of our horticulture employees. So the U.S. Botanic Garden orchid collection is about 3,000 specimens, around 2,000 taxa. Nearly 200 of those are vulnerable to extinction. And our key collections are in Bulbophyllum, Cattleya hybrids, and Paphia petalum. This is just a beautiful Stanhopea here that I love. And then the story that I want to tell you, though, about the orchid collection is this vanilla orchid. So we purchased this vanilla orchid from a grower and started growing it up this wall in the first room in our conservatory that visitors come in the garden court, really prominent location for us. And we wanted to grow this to get it to bloom so that we could really interpret vanilla to the public. And after about 4 or 5 years growing up this wall, it finally bloomed. And we were able to, collect to collect the stigmas from the plumea from the flowers and hand pollinate them to get vanilla beans. And at a previous job that I had at Brooklyn Botanic Garden, I actually would process these beans to make vanilla and plan to do that as well with these plants. And so the first year we harvested them to make vanilla and I processed them and did a few kind of maybe non-traditional ways of processing them. Here you can see they're on the dashboard of my car as I was driving across the country for a summer vacation. But nonetheless, all of my efforts failed and I did not get these vanilla beans to make vanillin. They would not smell like vanilla. There was no vanillin that was being produced. And so I tried again the next year and it didn't work again. And I had been very successful in doing this previously. So I thought, I think there's a problem here. So this is the plant in bloom. And this is Vanilla planifolia. Vanilla planifolia is the vanilla of commerce. It's where we get the vanilla extract that we all enjoy. And you can see here very clearly, I think all of you can see, this is not the same plant. And so what I did is I asked our orchid specialist to identify this plant. And in fact, it was not Vanilla planifolia. The grower that we had gotten it from had made an error in the identification, had sold us this plant, which is a Brazilian species that is not known to produce vanillin. And it's native to South America, French Guiana to Argentina. So we had an entirely different plant that we were growing as vanilla. We have relabeled this plant, and subsequently we have procured the proper Vanilla planifolia to be able to grow vanilla. But this is just kind of one little example of the importance of taxonomy and the expertise needed to identify plants correctly. So our taxonomic knowledge isn't just used to make sure that we're displaying the right things. It's often used to help us identify plants that come into our collection in various ways, whether that we're purchasing them, or more commonly, if they're donated to us or if we're collecting them in the wild. We get a lot of plants also through exchanges with other gardens as well. So we always are looking to duplicate our collections at multiple gardens in order to increase the sort of conservation value of those collections and ensure that if something happened to our collection, that the germplasm wouldn't be lost permanently. And another way that we get plants into our collection is through the CITES Plant Rescue Centers, through the CITES program as a plant rescue center. And this CITES is the Commission on the International Trade of Endangered Species of Flora and Fauna. And the enforcers of CITES in the United States is the U.S. Fish and Wildlife Service. And U.S. Fish and Wildlife Service has established a system of plant rescue centers, and also their animal rescue centers that zoos and aquaria participate in. There are around 60 public gardens in the United States that are rescue centers. And essentially what we do is we agree to take in plant material that's seized at US borders that's been brought into the country illegally. We have served as a plant rescue center here at the US Botanic Garden since 1986. And since that time, we've taken in hundreds of plants, mostly orchids and succulent plants. Just as an example, a single shipment that we received was all 99 species or 99 specimens of Dendrobium lindleyi that were believed to have been illegally taken from the wild and were being brought into the country under an incorrect name. The inspector at the border recognized these plants as likely having been pulled from the wild and was suspicious of their identification. And so they seized them and then asked the U.S. Botanic Garden to take them into our collection. We did so, and it took our skilled horticulturists about 2 years to nurse these plants fully back to health and to get them to bloom. And that, at that time, is when we were able to identify them definitively as Dendrobium lindleyi. They've been in our collection ever since. We're also involved in some communications around building education and awareness of the sort of concerns of illegal trafficking of plants throughout the world. So we're part of a steering committee with Botanic Gardens International on developing international communication campaign on the illegal plant trafficking. And you'll see that coming out hopefully later this year. We would not have been able to identify the Dendrobium lindleyi without our taxonomic expertise, but we also wouldn't have been able to identify them without our horticultural expertise. Those plants came to us in really poor condition. We had to nurse them back to health, as I mentioned. And without those skills, those plants would have died and we wouldn't have known their identification. And that identification is also provided to Fish and Wildlife for any possible resulting legal actions that might be taken against the people that were bringing them into the country. So horticultural science is an extremely important skill set that we have at public gardens and that we can offer to the field of taxonomy. We have skills in propagation, breeding, cultivation, and those can be applied to conservation and horticulture and are really essential in education as well. We propagate taxa to both build our ex situ collections, but also to contribute potentially those plants back into the wild for reintroductions. There are huge mutual benefits in utilizing horticultural skills to apply it to taxonomy and certainly to conservation as well. Public engagement is also really enhanced when we are displaying plants through the context of scientific knowledge. And by this, I mean that we are providing scientific information with our displays. We're interpreting our displays through that science lens as well. A big aspect of science at public gardens is collaboration. And I think that's true for all of science, really. But at public gardens, we have a huge skill set in horticultural knowledge. We often have wonderful facilities. We often have scientists on staff as well. But sometimes we don't have access to the populations that we want to study, or we need to work with other organizations to advance our missions to learn more, to gain more knowledge about the plants that we're interested in studying. Some of the partnerships that we've pursued here at the U.S. Botanic Garden are really in collaboration with other public gardens, with academic institutions, and with science and conservation organizations around the world. I'm going to talk about a few of those, and then I'll kind of go into a deeper dive and tell some more stories about some of them in particular. The Global Genome Initiative for Gardens was started at Smithsonian Institution and was really intended to collect all of the genera of plants globally and to put them into collections to allow scientists in the future to access that germplasm for research. And there's been great success in this program. It's involved gardens really all over the world, and there are tens of thousands of collections that are now in germ banks and herbaria available to scientific research thanks to this initiative. The U.S. Botanic Garden has partnered with the Flora of North America to improve the website of FNA and to help support the publication of the remaining volumes of FNA. We also partner with Botanic Gardens Conservation International and have produced a wide array of conservation assessments, both here in the United States, but also around the world, to look at some of the gaps that are in living collections, and also looking at particular species and the needs that those species have for both conservation in the wild and, if necessary, conservation ex situ as well. We have several collaborations with the Morton Arboretum, one of which is on the Global Conservation Consortium for Oaks. Global Conservation Consortia is our sort of initiative of Botanic Gardens Conservation International that brings together multiple organizations to advance the conservation of particular groups of plants. It may be a genus, such as is the case for the Oak Consortium and the Magnolia Consortium, or it may be a large group of plants, such as the case for the Global Conservation Consortium on Food Plants that we've partnered with the New York Botanical Garden and others to establish. The Global Conservation Consortium for Oaks has resulted in some really wonderful discoveries, again, led by the Morton Arboretum in partnership also with U.S. Botanic Garden. And this particular project that I'll talk about was also in collaboration with NatureServe and other organizations as well. This collaboration has located a plant that was previously thought to be extinct in the wild. So Quercus tardifolia, it's from the Southwest US. And this is the plant that there were historic records of it, but it had not been seen for decades. A group of the collaborators went into the field and were able to relocate it. And this plant is now in propagation for conservation ex situ, with longer term goals of getting this plant back into the wild. But what's interesting about this project too is that there's a lot of really interesting taxonomic questions here. We really aren't 100% sure that Quercus tardifolia is a species. It's probably not. We know definitely the ones that we found are of hybrid origin, and so there's a lot more work to be done with this to really determine the sort of taxonomic identification of these plants that we're now cultivating. But regardless of the final status of whether or not it is a species fully distinct from others, it's a really important plant for us to conserve in that it's located in this one area in the Southwest, as I said, but it also has a really good story. And I think sometimes when we think about plant conservation, we're certainly focusing on the importance of conserving that plant or that organism for the value that it brings to the environment, but also, of course, for its intrinsic value as a species. And there's a sort of a third category that I think is important from a broader context within conservation, and that's looking at how can that species help us tell a broader story of the importance of conservation. And sometimes, such as with Quercus tardifolia, people get really excited about just the rediscovery of it. So this is something that we thought was extinct. We found it again, and it's gotten people just so excited. In fact, the original stories that we worked with media on to get out there to the public, that original rediscovery of Quercus tardifolia, the media for that had a reach of over 1.5 billion people. And so really had a huge impact in getting people to think about plant conservation. So even if Quercus tardifolia isn't a plant in their backyard, it helps them understand that scientists are out there doing this wonderful research, doing this wonderful work, rediscovering things, and gets people excited about plants. Another partnership highlight that I want to talk about is the Center for Plant Conservation. And I don't want to steal Wes's thunder because he's going to be giving a talk next month for the same series. But we work with Center for Plant Conservation on rare species assessment and monitoring, on a little bit on habitat restoration, and on plant propagation and population augmentation. So kind of reintroducing plants that were grown ex situ back into the wild. So this partnership is both ex situ and in situ and relies heavily on taxonomic expertise. Another partnership that really relies heavily on taxonomic expertise is our collaboration with NatureServe. And NatureServe is a nonprofit organization that provides scientific information and tools needed to achieve conservation in North America. And you all probably know it as the organization that releases the conservation assessments for species. And so if you know through the ranking of a species in the United States, that was generated through NatureServe's process. So one of the part of the partnership that we have with NatureServe has identified 156 continental US and Canadian endemic plant taxa that either occur in only one location or only one location remains. Of these 156 taxa, 15 are listed as endangered, and endangered or threatened under the U.S. Endangered Species Act. 30 had NatureServe or IUCN red list assessments older than 10 years. So these are imperiled taxa that we really had old data about and needed to update the assessments of those taxa. The current status of this project is that we are beginning to advance conservation strategies for sites supporting a single site endemic taxon or taxon of one known occurrence. Conservation assessments considered out of date or older than 10 years have been prioritized for rank review. And we are funding collection of priority single site endemics or organisms of one known occurrence to bring them into ex situ collections. All right, so I've talked a little bit about science at public gardens. I think many of the sort of types of science that we do at gardens, I'm sure you're familiar with, as I said. And I want to talk a little bit about sort of how do we use our scientific knowledge, the work we do within our scientific partnerships to inform our displays and exhibits and engage the public and teach them more about plants and taxonomy. So some of our displays are arranged based on sort of the types of plants. So this is our desert collection. So it's an ecologically based collection, but it's not geographically arranged, anything like that. It's succulent plants from all over, all arid environments around the world. Some of them are really about the kind of type of plant. So this is our carnivorous plant display, one of our many carnivorous plant displays. We also have an outdoor bog display. This is actually a garden that was put in as a collaboration with the Atlanta Botanical Garden as part of the American Public Gardens Association meeting in 2019. And it's still flourishing today. You can see it's part of our rain garden system in Bartholdi Gardens, which is our southern garden here at the U.S. Botanic Garden. And it will hold a lot of rainwater when we have large storms. This is what it looked like yesterday when I was walking through the garden. And even though it's very winter state here, we can see the plants above ground are looking dead to many visitors, but they'll be turning green and coming back up very soon. But the plant labels are still there. And so I often see visitors just engaging with the plant labels, looking at what plants look like in multiple seasons and learning information about them from the labels. And I'll say that one of the primary ways that public gardens advance taxonomic knowledge in the general public is by labeling our displays. So making sure that we have the scientific name front and center. The scientific name is almost always the largest name on the label. So it's the one that draws the greatest attention. But you can see there's a lot of other information on this label as well. So we have not just the scientific name, but the common name, its distribution, Also, the family name is on there, and what we know about the conservation status. So the Sarracenia is a vulnerable species according to IUCN. The silver tag that you can see there is our accessioned tag. And that, as I mentioned, our living collection is an accessioned living collection, like any museum collection would be. And that label has lots more information about where we got the plant, when it came into the collection, that sort of thing. Sometimes our displays, though, are just really cool plants that are just charismatic and really get the attention of the public. But why do we grow? Why are we growing corpse flower? It's not a native plant. It takes a lot of space. They're really kind of finicky. So why do we grow this? We grow it because it is really an awesome, like as I mentioned with the Quercus tardifolia, an awesome sort of way to get people who are excited about a particular plant, a particular discovery, excited about those things, get them to kind of go to the next level with their knowledge about those organisms. So we take advantage of the attractive stinkiness of that plant and the many thousands, tens of thousands of people that come to see it by then, while they're here, teaching them about the plant more. We also use the corpse flower as an example within our teacher professional developments that we offer as well. So it's a fun kind of case study for people to understand more about a plant and understand that this is a plant that's also imperiled in its native environment, and kind of the work that we do at public gardens to both conserve that plant and teach people about it. So this is a life cycle of corpse flower that we actually developed with collaboration with a botanical illustrator. This is one that we put on display every time we have one bloom. But you'll also see this in the media. Other gardens use this. As a federal entity, the U.S. Botanic Garden will actually give any other public gardens the materials that we develop for their use in their educational programs. And this one has made the round. So you'll see it in media a lot when other gardens have one blooming as well. People are just fascinated by the corpse flower. They love its stink, its size. They love the ephemeral nature of it. You've got to run out to go see it because that bloom is only going to last a day, and it's really only going to smell the night of and the morning of the bloom. But we also use our corpse flowers for conservation work and scientific research. So we don't put all of them on display. You can see on the left here, here's one that we did put on display. And on the right, what we've got is one that bloomed at our production facility. And we did various experiments on it while it was out there, including looking at taking detailed photographs of the flowers and looking at the thermal energy that's generated by the corpse flowers. So part of that corpse flower research is developing thermal images. We also do a lot of cultivation experimentation, looking at different environmental conditions, different light conditions, et cetera, and how they impact the corpse flower. And we also collect pollen from many of our blooms to use for pollinating our own plants, but also sharing those with other public gardens and universities for them to pollinate their plants. And then the resulting fruits that we get will grow up into seedlings, and then we'll share those seedlings out with other gardens as well. We're also one of the gardens that participated in a research project led at Chicago Botanic Garden in looking at the genetic diversity of corpse flowers. So we contributed some tissue to that study, which really had the purpose of looking at the diversity of ex situ corpse flowers in both the US and European collections, and then being able to use that information to really inform how we breed these plants. So looking at the parentage, the lineage, the diversities within these collections, and identifying which of the plants that we should collect the pollen from to pollinate others, to maintain or increase the genetic diversity in cultivated collections. We've also contributed our corpse flowers to make herbarium specimens. So this is a group from Natural History Museum here in DC at the U.S. National Herbarium, making some specimens and actually also collecting some tissue for cryopreservation and silica gel preservation for future research as well. One of the interesting things that we have been able to document in our collection is the thermogenesis of the inflorescence. And so here we have a photo of the thermogenesis of the spike there of the spadix. The spadix of the spike inflorescence. And what you can see is that that's the top of the spadix there is really heating up. And this is what we see at female maturity. So when the females are receptive to pollination, we see a massive heating of the spadix. At male maturation, which is about 12 hours later, we see the same thing, but it's lower down where the heat is generated. And that is the sort of indication that the pollen is about to be shed. And you can see it shedding there from the anthers on the right. This is just kind of a side-by-side showing two thermal images of the same inflorescence. This one was on display to the public, and we took images overnight and in the next morning. So you can see just before midnight, the female flowers were receptive, the spadix started heating up, and then 12 hours later, the lower part of the spadix was heating up when the male flowers were releasing their pollen. Our Amorphophallus collection, though, doesn't just start with Amorphophallus titanum. We have over around 78 Amorphophallus taxa in the USBG Living Collection. And we are actively establishing a global garden-based cooperative working group to conserve and study this genus, with the idea of creating what's called a meta-collection. So looking at sort of a multi-institution, in this case, multi-country as well, collection of Amorphophallus in ex situ collections to advance the conservation and knowledge of Amorphophallus. So the corpse flower is more than just a display. It's really just one little way that we can bring the public in who are excited to see this stinky plant. And then while we have them, we can educate them about the diversity of the genus. We can educate them about the wonderful research that's being done at public gardens to conserve this plant and the threats to this plant in its wild populations. If you want to learn more, you can go to that QR code. So how do we then use our displays to engage people and teach people more about plants and also about plant taxonomy? Well, botanical training is really at the core of so many public gardens here in the United States and really around the world. And as I said earlier, that education really starts with pre-K and it really goes through retirement. Many public gardens have certificates that they offer in botany, and many of them partner with universities to offer graduate degrees. And those programs really, as we've seen academic institutions change their departments to be more sort of evolutionary based versus organismal based, or broader sort of biological sciences and whatnot, what we've seen is a shift really to where taxonomic training is happening, to being used to be more primarily at universities, with certainly some public gardens engaged. And there's really been a decrease in some of that taxonomic training at universities and a shifting to botanic gardens. So I would say that botanic gardens and their role in educating the public, but also educating future plant taxonomists has really increased over the last 20, 30 years. And this education is done through just simply some of the displays that we have, through engaging opportunities within exhibits, and through direct engagement with the public. We like to greet our visitors with discovery carts in the garden court, where we have our volunteer corps that's been trained to talk about a particular group of plants or a particular suite of plants within our collections, whether that's orchids or gymnosperms, or might just be talking about chocolate, one single species in our collection. But we also bring in other experts as well, such as botanical illustrators who are very trained in slow looking and looking really carefully and methodically at plants and reproducing their structures. So the two pictures on the right there are two different botanical illustrators that came in and offered their expertise to the public on how to really reproduce with scientific accuracy plant structures. But science education at gardens doesn't just look like talking to experts and reading signs. Sometimes we, as I said, we like to, when we have somebody here, we like to take advantage of that and to go ahead and teach them about science, even if their visit wasn't focused on coming to learn about science. So this is a picture of our holiday show. We have a train show that we do every year, but the theme of that train show is always connected to our mission and connected to plant science. So this year's theme that I'm showing here from a few years ago is international agriculture. And every exhibit always features these wonderful little structures that are made out of plants. So the theme of international agriculture enabled us to talk about the importance of food plants, the diversity of food plants, where our food comes from, how we grow food. But the structures themselves, these little models themselves, give us an opportunity to talk to people about the plant structures that were used to make those models. And so we have this wonderful discovery cart that's really focused on how these models are made. And it has a sort of a palette of different materials that are used that are all plant parts. And we get to talk to people about the identification of those plant parts, what plants they came from, what the structure names are for those, so the morphological terminology associated with it, and then, of course, the building process for the models themselves. It used to be that we had our train show inside. As you can see now, it's outside. Outside is great because there's no lines. There used to be long lines to come into the train show. And when we had those long lines, we thought, wow, this is another opportunity where we can engage people about plants while they're waiting to see trains. So these are just a couple of the signs that were from when we used to have a long line to get into the train show. That the year these signs are from, the theme was pollinators. And so we had these more sort of deeper dive informational signs into about pollinators and about the pollinators that are featured in the exhibit. And I'll say too, in all of our interpretation, we're always identifying the scientific names of plants, so that we're always constantly using the taxonomy of the plants. We also have a captive audience in our bathrooms. And so on the back doors of the stalls of our bathrooms, and I've seen this at other gardens too, this isn't unique to the US Botanic Garden, we have interpretive signs. Some gardens you'll see, sort of what's happening at the garden today, because you're a captive audience there and you're probably gonna read that sign. So we have poop themed signs here at US Botanic Garden that we use to engage the public about plants and their connection to poo. So you're probably familiar with some of these stories, but I love the shrew here on the Nepenthes, using the Nepenthes as a source of nectar, licking the hood of the Nepenthes there, the pitcher there, and then while it's there, using the bathroom into the pitcher. And the pitcher, of course, uses that as a source of fertilizer. So it's always excellent to take advantage of opportunities when you have visitors that are going to read your signs. We develop large exhibits every 2 years here at US Botanic Garden. We have a gallery space that we put these exhibits in. And these exhibits are really focused usually on a plant science theme, or certainly if it's a broader theme, there's a lot of plant science stories within that space. So this is one of my favorite exhibits that we've done. It was about 10 years ago. We did this exhibit called Exposed: The Secret Life of Roots. And these are plants that were grown at the Land Institute in sort of baked clay. And so that they could actually be there in these long columns, and they could be removed from that and still show their full root mass. And so the plants you're seeing here, these are all native prairie plants. And these native prairie plants, as you can see, have very long root masses. They were so long that we had to sort of loop them back and tie them up, even though they were hung about 10 feet off the ground. And this was a wonderful exhibit that gave us an opportunity to really explore the underground structures of plants. And we had a lot of interpretation in the exhibit, really teaching people about plant morphological terminology associated with these underground structures. And so we had some quizzes, true root or imposter, where we got to talk to people about and teach people about the underground structures that are not really roots, the stems that are underground, maybe as storage or sort of plants that are growing horizontally underground and whatnot. Our current exhibit in our gallery is called Fierce Flora: Tales of Survival and Demise. And that exhibit is up through next year, and it's been really well received. It's a wonderful exhibit that really talks about plants that are kind of wicked in some way or another, like they have spines or thorns, or prickles, they're poisonous, they're carnivorous, they're parasitic, etc. So lots of really wonderful stories told in this gallery. There's several selfie stations that are featured that show plant morphological structures that are important in identifying some of these fierce plants. So this is my favorite panel, of course, Toxicodendron and radicans, pictures of the fruits, the flowers, the adventitious roots, and the leaves in different seasons. There's also a little herbarium in this exhibit, and it features some tactile herbarium specimens that we had made, where they're specimens that are pulled from the images were pulled from multiple different herbaria. Quite a few of them are from the New York Botanical Garden. And then we worked with an artist that does tactile printing for low vision and blind people to create these so that the public could really engage with these herbarium specimens. And I'm happy to say that we reached out to the New York Botanical Garden about this when we were doing it, just to say, hey, we're reproducing a bunch of your specimens. So, is that all right? And they were so excited about it that they came down to see the exhibit, and they've actually made some of these models themselves. So when they're giving herbarium tours, people can engage with the specimens in a way that they can't otherwise. We also sort of coincidentally featured a specimen of Anacardium occidentale, the cashew, that was annotated by John Mitchell, who is a scientist at the New York Botanical Garden. And he came for a visit, and I was able to show him his specimen and his annotation there, reproduced in a tactile herbarium specimen. But we don't just engage people with sort of exhibits for them to read and read signs. We know that some people don't really read signs, or might not go through and read the interpretation. So we do a lot of direct engagement with people as well here at the U.S. Botanic Garden, but also really at public gardens globally. Again, taking advantage of that large visitorship. We do this with our own in-house expertise, but we also bring in other experts as well. So we have a partnership with AAAS that brings in plant scientists that are associated with AAAS to engage with the public on their particular areas of expertise. We also engage with the media. Corpse flower coverage alone is just an enormous outlet for us to really engage a global audience in plant science. So just this one bloom we had last year had a reach of over 890 million people. It was covered in 332 international articles. And we have videos online that we've posted about the corpse flower, which have garnered millions and millions of views as well. So what's the future of science at gardens look like? Well, I think one thing that we need to do is just create more collaborations. I talked a lot about the partnerships that we have here at US Botanic Garden today, but really looking to collaborate with universities, other gardens, conservation organizations. We need to continue ensuring the duplication of living collections to preserve germplasm for the survival of species and for future scientific research. We need to make sure that we're sharing knowledge with each other so that those best propagation practices, et cetera, can be realized at other gardens as well. And one important area is also increasing tropical collections. They are sort of disproportionately underrepresented in gardens as they are in herbaria. So this is something that needs to be a focus for sure moving forward. And I want to give a shameless sort of shout out to a botanical symposium that we help organize with Smithsonian every year. This year's theme is American Botany, 250 Years of Discovery. This is a half-day symposium. You can see the speaker list there this year. Andrew is one of the speakers that's going to be in that symposium. We're super excited about that. You can go to that QR code to register, and it's free and online. It's a hybrid event. If you have any questions for me, you can reach me. This is my email address, susan.pell@aoc.gov. And you can also follow the US Botanic Garden on social media. And then I will take any questions you all have.
Susan, I just want to first say thank you. That was a fantastic talk. Really exciting to see how US Botanical Garden is weaving both research and particularly taxonomy through so much of the interpretation and exhibits you do. So thank you very much. And I'll open it. Catherine will moderate the question and answers.
Thanks, Susan. So our first question is from Dwayne Coulterman. Are you in contact with the project to reestablish vanilla as a cash crop in Puerto Rico?
Ooh, good question. We are not. Yeah, we haven't done really any vanilla research. There was a scientist at New York Botanical Garden that was doing some vanilla research for a while, but not really related to propagation, but love that. That sounds like a great idea.
Lots of comments thanking, saying that they're really enjoying the webinars. We're delighted to hear that. From Jesper Axelson, what is micropropagation if you would describe it to someone who doesn't have much knowledge on plant science?
Oh, fantastic. So micropropagation, usually what we mean by that is tissue culture. And so taking small plugs of plants, typically it's leaf tissue, and then growing them on media, typically like an agar, kind of a gel media, up until they're sort of large enough to then transfer into a regular media, so soil-based media. So yeah, that's typically what it is. It also can be other things too, but typically we're talking about plant tissue culture.
From an attendee, any suggestions on bridging the gap between horticulture industry, botanical conservation, and hobbyist communities?
Ooh, and hobbyist communities. Yeah, so I think there's I give quite a few talks for like master gardener groups. And that's really common for a public garden world to really interface a lot with master gardeners. And of course, extension offices also are where a lot of master gardener programs are based. And so that's a really nice connection with academia as well, especially at our land grant universities. I do think, though, that there's quite a few conferences that I think would be really of interest to the general public too, if they sort of knew more about it. Maybe things like the American Public Gardens Association conference, where there's a diversity of kind of different sessions. I mean, I think the botany conference would be interesting to people too, but maybe not the broad general audience. I think that the interface between academic institutions and the scientists at public gardens and the public facing side of public gardens is something that I've seen kind of expand over my career. So the sort of public gardens used to be, I think, and some still potentially are, kind of siloed within their organization, so that they're sort of, science is maybe not talking to the exhibits people. And I've really seen a breakdown in those silos kind of over my career. And I think the public interest in science is so strong, and so it's really important, I think, just even from if you're just looking at it from a numbers perspective, to really engage your exhibits team, your education team, with your scientists. And here at US Botanic Garden, we really kind of have a science lens that we use for pretty much all of our programming.
Next is from Jennifer Blanchard at the Woodlands Conservancy in Belle Chasse, Louisiana.
Nice.
She says that it's not a botanic garden, but it's an organization dedicated to the protection and restoration of coastal forests. And she's interested in how to best begin partnerships to expand research on the site. Can you give your insight on how to expand engagement with the site?
Yeah, so I will say, well, so one, that the American Public Gardens Association a few years ago really updated their kind of definition of public gardens. And so probably you might be a public garden, even if you think you're not, which is fun. So there's lots of things, some community gardens are really public gardens. A lot of university campuses are public gardens. Public parks and whatnot can be public gardens. So don't sell yourself short on that. But I think, kind of identifying what your local community's needs are is one way to really engage people. Something that we're doing at USBG that I didn't mention, it's not super science focused, is a community engagement effort where we're really doing listening sessions, working first with community partner organizations. So organizations that are already in your communities, that are supporting your communities, talk to those people and ask them, what does the community need? And how can we align their needs with our existing mission? So you don't want to bend yourself too much to kind of reach an audience. You want to find where those synergies already are, and be able to kind of meet the community where they are, whether that's literally going to the community and meeting them first so that they know they're welcome to your space and want to come there, or whether it's just connecting with the community organizations and getting the word out about your organization.
Just as a note, we threw a link to the registration for the National Botanical Symposium in the chat.
Fantastic. Thank you.
And I think we have time for maybe one more question. Dwayne, I'm going to see if we can come back to yours for a clarification, because Mason McNair has a great question about the tactile herbarium specimens. Are these models available for others to download or order anywhere? My lab has been developing 3D printed educational models and have had very little luck finding good ones online. Yeah,
so great idea. So these are, the specimens are not, I think, not technically 3D printed. It's sort of a different process. But I would be happy to share like who we've worked with and the specimen kind of list that we have already. You sort of have to create a kind of a detailed design file. We can share the design files. Happy to share design files, but it does take a skilled graphic designer to be able to create those files for the reproduction of the specimens.
Okay. Let's see. I think that may be all the time we have for today. That we have time for more?
I want to say
Susan?
thank you so much. And also, I'm
Yeah.
so sorry for having a strange health crisis at the first half of my talk. So
Not
apologies
at
for that.
all, Susan. We're
Yeah.
just glad you were okay.
Yeah.
Selfishly, because we would have missed seeing the second half of your talk. So let's give a big round of applause for Susan. Thank you very much to all of you who took time to attend today. We really appreciate your time. This is like this community that we're in today. This is a really big and an important part of ASPT for us. It's a part of a great deal of what we want to accomplish. We're grateful to you all for being here. We hope to see you again next time. Susan, thank you again for a fantastic talk.